This is part two of our lecture, recorded lecture on Module 3, Microorganisms in Industrial Microbiology and Biotechnology. So we will cover the remaining discussion on culture media. So there are culture media which are both selective and differential at the same time. So they can be used to distinguish microbial colonies of the target organism and at the same time inhibit the growth of those that we don't want to isolate. No? So some examples include mannitol salt agar, which is encouraging or capable of encouraging the growth of um, staphylococci, mostly staphylococci, while inhibiting the growth of others. There's eosin methylene blue agar, which is useful for gram negative bacterial isolation. It will inhibit the growth of gram positives. There is TCBS, thiosulfate citrate bile salts, selective for the growth of Vibrio and can also be differential um, across the different species within the Vibrio genus. We also have triple sugar iron, which is an agar medium that allows you to identify and differentiate bacteria based on their ability to make use of three different sugars and at the same time release acid and hydrogen sulfide. Then there's bismuth sulfite agar, which, if I remember correctly, is a selective medium for Salmonella species. Okay, right. For anaerobic microbes, these media, as the name implies, will contain or does not contain oxygen, little or no supply of dissolved oxygen at all. They may have reducing agents that are auto-oxidizable with great affinity to oxygen, so we can identify which portions of the medium actually contain trace amounts of oxygen. Also, the most anaerobic media are semi-solid in appearance. Will decrease and the reason behind that is to ensure that oxygen doesn't really diffuse through the depth of the medium, through the vertical depth of the medium. Bacteria will grow in the medium according to their requirements, their respiratory requirements, whether they are requiring oxygen or not. So, thioglycolate fluid medium is an example of a medium that has an area that contains oxygen and in a section that also lacks oxygen. Okay. Um, right. So, in general, there are several principles, or not several, but there are generic principles we follow uh, when we want to grow and examine microbes in the laboratory, especially in the context that we are investigating them for their potential industrial applications. Okay? So, five things, five I's. Inoculation, which is the introduction of a small amount of the sample, an inoculum, into a nutrient medium. Incubation, which is when we create the condition for proper atmosphere or ambient for the proper growth of our target microbes. So usually we manipulate or control the temperature during incubation. And in some instances, especially if the target is anaerobic microbe, an anaerobic species, we also have to control the presence of oxygen. Then we have isolation, which is separating a bacterial cell, a single bacterial cell from others, isolation or purification. Then we inspect what grew, or how was the growth, how what organisms grew after we did our first three eyes. And lastly, of course, most importantly, we identify the microbe. We are able to assign a specific taxon. No, we identify what species or what genus the microbe belongs to. Okay, so a review on bacterial growth phases. So in any setting, bacteria grow following this uh, standard, standard growth phase, not really spectrum, but graph, no, growth rate graph. So we have four major phases of bacterial growth. So we have the lag phase, where the microbe will carry out intense activity in order for it to prepare for cell division. um, it will sense the environment if there are proper nutrients, or the nutrients for its growth are available in the ambient. Will adapt to the general conditions of the ambient. Once the microbe has successfully sensed the environment, it will now begin exponential increase in population, in cell number. Then eventually a saturation point will be achieved, the stationary phase, where there is period of, or which is described as a period of equilibrium. So microbial cell death is equivalent to microbial cell reproduction. So the rates of cell division and cell death are the same. Then lastly, bacteria can eventually a bacterial colony will die, population will decrease at a logarithmic rate, um, due to several reasons, like a change in the ambient factors, like change in pH, temperature, oxygen availability. In most cases, in wet laboratory, when when the culture medium has been exhausted, no nutrients available anymore, and there's no addition of new portions, or new available concentrations of nutrients, then the bacterial cells will begin to die. No, usually the that phase also marks the stage in which bacterial cells, for the case of those that produce resistant spores or endospores, will begin, no? Or will start. The sporulation step or sporulation stage may begin. Okay. All right. Then, okay. Now let's look into the last section of our lecture, application of microbes in biotechnology. Okay. So in our laboratory, in our first activity in the lab, we are able to appreciate the diverse set of microbes from the environment and other types of sources, other types of reservoirs, where we can potentially get industrially significant microbes. So we exploit them after um, after we are able to identify the taxa or give it a taxonomic assignment. We look into many things in order to optimize the microbes cellular machinery in terms of producing our product of interest. Okay, so for example, some microbes have been genetically engineered for the production of human insulin. Others have been used widely in the food industry, in the food and beverage, as well as in agriculture industry, for their capacity to ferment uh, uh, macromolecules, or ferment large molecules. Some of them have been utilized for the production of biofuels. Others, in terms of the in terms of crime scene investigation, as well as human security concerns. And lastly, or another example of how we exploited microbes to our benefit is the creation of or the production of medically significant products like vaccines and antibiotics from microbes or using microbial systems. So in agriculture and food industry, we have improved crop yields ever since we discovered that we can actually make use of microbes to introduce genes or to carry out heterologous gene expression. No, so we can introduce genes that can confer resistance to insects, tolerance to pesticides, and resistance to extreme environmental stresses such as too much temperature, lack of water, no, or too little temperature or too low of a temperature, no. A good example of this, I believe I mentioned this last Saturday, the Bacillus thuringiensis bacteria, which is able to confer resistance to pests when they are used as biopesticides, no. Then, in terms of food, I cannot emphasize this more. Microorganisms as major fermenters of food products. So we make use of these microbes to ferment our food products, and also at the same time, no, the process yields bioproducts that are useful to us, like the ones that are shown here. So you can see different genera of bacteria and what they produce as they break down pyruvate. No? Some can produce mixed acids, others produce acetic acid, alcohol, butyric acid, okay, propionic acid. So these are just examples, some of the few examples of fermentation products that we get from microbes. No? Now, other products that we get from microorganisms, we typically divide them into two types, no? A primary product or a primary metabolite, and a secondary product, secondary metabolite. A primary metabolite is a major compound that is essential for the for the continued development and growth of the microbe. So some examples include building blocks of proteins, amino acids, some vitamins and alcohol, as well as organic acids. Secondary metabolites, on the other hand, they are produced or bioproduced by microbes in response to a change in their environment. Like if there's competition, they might produce antibiotic compounds that inhibit the growth of other microbes within or microbes occupying the same space that they do. Others also produce pigments that we make use as food coloring. Others may produce toxins depending on how harsh the environment is, so that they increase their chances of survival, they increase their chances of dominating the community where they are at, and maximizing the availability of nutrients that are present. No? And there are mostly enzymes. So enzymes, in a way, are I would say in the midway between or in the mid type between a primary and a secondary metabolite. So these enzymes are important for breaking down products, for producing products, and also for carrying out related processes that ensure cell survivability over different types of stressors. Okay, or during the presence or in the presence of different types of stressors. So some examples of enzymes that we harvests from microbes and we make use of are the following. So lipases for cheese making, lactase, which breaks down lactose to glucose, and galactose for the production of lactose-free milk. So individuals that are lactose intolerant, they can still consume milk as long as it's lactose-free and thanks to the lactase enzyme. Then we have proteases that can serve as laundry additives for the breakdown of proteins. Then pectinases, which can degrade pectin into more soluble components. No? So the material or the liquid medium will not be, or the liquid material will not be that cloudy for the case of wine and fruit juice. Pectinases are added to homogenize the appearance of the liquid material. Okay. Then we have for energy production, for medicine. So we have the following examples. For bioethanol production, bioethanol fermentation, so plant starch, cellulose from agricultural waste, and generally garbage or commercial garbage. They are abundant and they can serve as substrate material for bioethanol fermentation. Then for the production of vaccines, okay, some microbes are used as expression systems, no, for the production of vaccines of different types, or vaccines mostly for viruses. Okay. And then in the field of law, legal, we have bacteria that can be utilized for crime scene investigation. If we're familiar of microbial forensics, okay, which is a field where we make use of microbes as clues, as evidence to describe or to get an account of what happened in a crime scene. No? Then for in the case of global health security, there are bacteria that can be used for bioterrorism. Okay, and we and these bacteria or these microbes, mostly the ones that have very high levels of pathogenicity and contagiousness. Okay. Um, so not really related here, but I guess more on the biotechnology aspect. So some the bacterium Thermus aquaticus has been utilized as a source for industrially significant enzyme, the Taq polymerase, for PCR applications, used to amplify biomarker, DNA fragment, no, a gene. So the Taq polymerase is heat resistant, so it is the optimal type of polymerase in artificial setting, no? Since PCR is essentially an in vitro setting for an in vitro process of DNA amplification. Okay, then we have, so I mentioned this already. I think I mentioned genetically modified organisms in the last meeting. So some examples. We have for the production of insulin. In the past, animals were used for insulin production, but since we discovered that we can actually do recombinant DNA technology, or we can apply recombinant DNA technology to produce medically significant products like insulin, then the manufacturing industry made use of microbial systems. No? Okay, so this one is just a visualization of how insulin is produced within a microbial system. So there is a vector, this one, your expression vector, that contains a bacterial promoter recognized by the bacterial cell, and then the gene of interest, okay, for this case, insulin A protein chain for expression. The same with the other half of the insulin protein, beta chain. Okay, so we transform our microbial system, in this case, an E. coli cell. We transform E. coli cells by inserting the vector inside them, and then eventually allowing them to express, or allowing the microbial system to express the protein, purifying the protein, and then eventually combining them so that they can become medically grade. They are so that it becomes medically grade or medical grade, and can be used for human application. Of course, antibiotics definitely. Um, Mostly, no, antibiotics have been sourced from microbes that are soil inhabiting and marine water inhabiting. So, in our example, or in our wet lab, probably we're able to observe or isolate some soil microbes like actinomycetes, which are known, really popularly known as producers of antibiotics. Then, in case of remediation, so we use microbes to remove contaminants, pollutants, and toxins from uh, fresh water and from soil. Okay, so there are some examples that I've already discussed last time. So I think this is the last slide for this presentation. So in our course pack, I added. Okay, let me check. In the course pack, I added a section for asynchronous reading, some examples of taxonomically, or some examples of industrially significant microbes that are identified um, based on, or that are identified that are identified and known to be industrially significant. So let's see here. So I added a section here, some of the known microbes, and please take time to read through the section. It's not very long, so just some examples and some um, emphasis on the concepts I've already talked about, especially on the key characteristics of industrially significant microbes, biotechnologically significant microbes. So there are a few on, there are some on some discussion or reading material on bacteria, on fungi, on algae, and archaeans. Okay, so that ends our Module 3 lecture. Please take time to watch the two-part video or the two-part discussion. Your enabling assessment short quiz for Module 2 and 3 will be activated this Saturday afternoon. You'll have until Tuesday to complete the enabling assessment.